all sorts of support. So some of them are actual support, like holding the neuron. Some of them are just sort of keeping other things away. Some are moving the cerebrospinal fluid. So there are 6 types, and they are all based on function. 4 of them are in the central nervous system. 2 of them are found in the periphery. So we'll start with the central nervous system first. We have the left-hand cells. These are known as astrocytes. So astrocytes, these are named because they look like stars. They have lots of arms, right? Stars in the sky, and they sparkle, and they look like they have rays. And that's why somebody named them an astrocyte. So the astrocyte is going to be close to the capillaries. The astrocytes help to create the blood-brain barrier. The blood-brain barrier. Now, this keeps unwanted things out of the brain. Keeps unwanted things out of the brain, which is good, except not every medication that we as human beings have made can cross the blood-brain barrier. And so specific medications have to be created to cross the blood-brain barrier. Many times certain chemotherapy drugs do not pass the blood-brain barrier very well, which is why many times if you wind up with a brain tumor, it is surgical intervention if you can, but that's why they use radiation, right, to try and get rid of the tumor. So that is our astrocyte. The other one, we've heard of these before. These are the ependymal cells. These are the cells with the cilia that are going to move the CSF. So this particular diagram comes from the central canal of the spinal cord. So it's lined with these ependymal cells that are moving the cerebrospinal fluid in and around the brain and the spinal cord. So those are our first 2. Then we have our microglial cells. The microglial cells, as the name implies, are tiny. These are tiny. The microglial cells, these are the cells that are very, very small. They are also phagocytic, which means they consume things. And so these are going to be wandering around in the brain, consuming things that are either broken or need to be gotten rid of. Think of them sort of like little vacuums, right? Wandering all over in the brain. These are the ones that, I think we talked about them last week or so, that the group, I think it's out in California, they were able to get in and do some gene therapy on the microglial cells so that they would target the beta amyloid plaques in the brain. Beta amyloid plaques are, for lack of a better word, bad in the brain. Beta amyloid plaques are going to contribute, they are one of the reasons or causes of Alzheimer's. And so they programmed these mice microglial cells to actually target the beta amyloid plaques, and they were successful. So as we talked about, this was what we call proof of concept. So it does work. It did work. And so now the question is, will it work on something besides a mouse? So hopefully it does. Then maybe we can start to eradicate Alzheimer's. But the other cell is known as an oligodendrocyte. The oligodendrocyte, oligo refers to many. Dendro refers to arms. So this is a many-armed cell. And what this does, if you look up here, here's the nucleus, this is the cell body right here, and it has lots of these arms coming off, or extensions, and they are wrapping around the axons. Multiple axons can be wrapped by one cell. The wrapping is known as the myelin sheath. The myelin sheath. This forms like an insulation around the axon. It also is going to allow the action potential to move faster. Faster, much, much faster. Up to 120 meters per second. So but the oligodendrocytes are only found in the central nervous system, brain, spinal cord. So these are the four in the central nervous system. We have our astrocyte, our ependymal cell, microglial cells, and the oligodendrocyte. In the periphery, we have two. We're going to start on the right. We have the cell that forms the myelin sheath in the periphery. It is known as a neurolemocyte. The neurolemocyte. Now, it also wraps around the axon, but it is different than the oligodendrocyte in that if you look at that right diagram, every cell there forms one section of that myelin sheath. So we would have one neurolemocyte wrapped around the axon. We would have another neurolemocyte wrapped around the axon, and another one, and another one, and another one. They don't touch, right? The neurolemocytes do not touch, nor do the oligodendrocyte. There are spaces in between, right? So if we look at this, each one of these is a separate cell. So we've got 1, 2, 3, 4, 5, 6, 7 cells. And in between each cell, there's a little space. It is called a node. They are the nodes of Ranvier. These are going to be very important when we go to move those action potentials down the axon. So again, the neurolemocyte creates the myelin sheath in the periphery, and each section of the sheath is one cell. The oligodendrocyte creates the myelin sheath in the central nervous system and can wrap around multiple axons and create multiple sections of the myelin sheath with one cell. But we can also see, we also have those nodes here. So there's a node of Ranvier, there's another one, there's another one, there's another one. Okay? So the oligodendrocyte is only in the central nervous system. The neurolemocyte is only in the periphery. Even though they both create the myelin sheath, they do it slightly differently. And then the last one are these little tiny pink cells. This is a ganglion, right? This is a ganglion. This is, we can see we have some cell bodies here. This is an axon. It happens to be myelinated. So every single one of these little white sections is a neurolemmocyte because we are in the periphery. And these cell bodies, the cell bodies are very fragile. They're very fragile. So in the ganglia, we surround them with these ganglionic gliocytes. They're also known as satellite cells for protection. Now, ganglionic gliocytes tells you exactly what they are, glial cells, and where they are, ganglionic, right? So, but they protect the cell bodies in the ganglia. Because again, the ganglia are basically outside of the central nervous system. Okay? Do these make sense? So we have four: the astrocyte, ependymal cell, microglial, and the oligodendrocyte in the central nervous system. The ganglionic gliocyte and the neurolemmocyte, they are in the periphery. Now, the neurolemmocyte and the oligodendrocyte, here, they are side by side, right? This is the oligodendrocyte wrapping around multiple axons all at the same time, creating a section of the myelin sheath. This is, each one of these is a neurolemmocyte, and you can see that it's a lot of wrapping. Now, inside the cell membrane of both the neurolemmocyte and the oligodendrocyte is what we call a lipoprotein. So this is a fat, a lipid, and a protein that are combined into one molecule. Now, what that means is that in our brain, our spinal cord, and our nerves, basically they're surrounded by a little bit of fat. And you will see that today in the sheep brain, especially when you go to clean things, because fat does not dissolve in water. You gotta use a little bit of soap, right? But it helps to protect the axon. Helps to protect, also helps that to move the action potential much, much faster down the nerves. So this is a picture, right? This is a picture. This is up close and personal of one axon. So this, right here, this is the axon. Right. So this is the axon. All of these layers right here, that's this right there. This is a neurolemmocyte wound around and around and around and around. And then this area right around the outside, here's a cell going all the way around. That would be the endoneurium. Remember when we talked about nerves? We said that the axons were surrounded by the connective tissue. We called it the endoneurium. That is there, whether there is myelin or not. Still have that endoneurium. So these basically have sort of like two layers of protection, right? But the myelin also really helps the impulses to go much, much, much faster. Now, one of the other things that myelin can help us do in the periphery is if a neuron gets busted, right? If you damage a neuron, right? Not the cell body, But if you damage the axon, that's going to be out in a nerve, right? You damage it, and it's, right, the rest of it basically dies, right? But the cell body is A-okay, and the stub of the axon is okay as well. If the axon was myelinated, there is the possibility of what we call regeneration. We're not doing mitosis, but what's happening is on the other side of the break, if those neurolemocytes are still functional, they can repair themselves and basically send out a chemical called chemotaxis, right? That is the process. We talked about this a little bit when we talked about inflammation. Remember mediators of inflammation? They went out and they triggered something to happen, like the neutrophils to show up, the macrophages to show up. Same principle. So those on the other side of the break, if those neurolemocytes are still functional, they will put out chemicals. Think of it sort of like a smell, right? And the axon detects that and can start to extend. Again, no mitosis. We're just extending the axon, and if we find those neurolemocytes, then can extend down the exact same pathway. Now, there's a lot of ifs to this. If the neurolemocytes aren't damaged beyond repair, if there is no scarring, if there is still part of the axon left out far enough. So again, there's a lot of ifs. But this is also why if someone, say, damages a nerve, after a while, after the inflammation has gone down, after things have healed, they may actually gain back a little bit of function because of this. Again, there's a lot of things that are iffy about this, but it is possible. It is possible. Now, we do get our myelin sheath. It's pretty much developed and ready to go by the time we are one. By the time we are one, we have our myelin sheath. Obviously, we're going to need to grow it with the rest of us as we get bigger. But this is also one of the things we like about myelin sheaths. You cannot do this with a neuron that does not have a myelin sheath. So we have two kinds of neurons, ones with myelin and ones without. So the only ones that have the limited regeneration would be the ones that have the myelin sheath. Now, how do we know which is which? Well, we can actually look at, we've seen this picture before, right? We can see the brain. The brain comes in how many colors? Two. What are they?
White and gray.
White and gray, yeah. So today in the sheep brain, it's gonna kind of be beige and dark beige, right? Because they have been soaking in a non-toxic preservative.
Yeah.
So, unmyelinated. If the neuron is unmyelinated, it is going to be gray. If the neuron is myelinated, it will show as white. So when we are looking at this cross section of the brain, all that is gray and even the dark gray, all of that is unmyelinated. All of that is unmyelinated. The center, all of this white, that is all myelinated. So it all has the oligodendrocytes in it. Now, again, in the brain, we know that the white is deep and the gray is the outside, or what we call the cortex. And in the spinal cord, the white is on the outside and the gray is in the center. So we actually have more white matter or white columns in the spinal cord. So basically things can move faster, right? They can move faster up and down. Now, in the periphery, we also find gray matter in the ganglia. Because the cell bodies are not myelinated. They just have those gliocytes around them. Okay? That makes sense.
Yeah.
Any questions?
I do got a question.
Yeah.
What's the reason why there isn't all just myelinated cells in the brain again?
Because there are some things that we don't need to move as fast as they do when they are myelinated. So when you send an action potential down a myelinated neuron, it has a specific speed, and it is around 120 meters per second.
Got it.
Okay? Unmyelinated neurons, where there are things that we don't necessarily need the action potentials to move that fast, we would want it to go slower. Hard to do on a myelinated. So unmyelinated, it's going to take a little bit longer, like 1 meter per second.
Okay.
Even so, most people are 2 meters or less in height. So if you're sending something from your toe to your brain, it's still going to take less than 2 seconds. I mean, if that's as far as you need to send it.
Got it.
Okay, other questions? So, we now know the basic parts, and so now we need to start looking at how does all of this work together. So we need to start looking at membrane potential. We have already seen that when we looked at muscles. We did introduce that. Remember, sodium on the outside, potassium on the inside. We got a whole bunch of channels in the membrane. Now we're going to see exactly how that gets set up and how we change it. We're also going to throw in a couple of numbers for voltage, because we know that anytime we move an ion, we create some electricity. So we actually can measure the electricity on our nerves. So what we are going to do is, here's our multipolar neuron. We are going to look and start at our resting membrane potential. We did define that when we looked at muscle cells. And then the stimulus produces what we call a graded potential. Now the graded potential, that's the thumbs up and the thumbs down. How many of these are coming in through dendrites, through synapses with other neurons, and they are all being sorted right here at that axon hillock. At the axon hillock. And if there are enough thumbs up, we can produce an action potential. So for us, we are mostly going to assume we have enough thumbs up, and we are going to create the action potential. We will discuss what happens when we get more thumbs down than thumbs up. But for the most part, we are assuming that we have enough. We are go for launch. Okay? And the action potential will then move down the axon. And then once we get to the axon terminal, we are going to look at synaptic activity or synaptic transmission. Again, we looked at that in the muscle system. We're going to add, I think, 1 or 2 little details, but you already know synaptic activity. So that's what we're going to do. So we're going to start by looking at the membrane potential and action potentials. How do we make them? How do we maintain them? And then how do we move the action potential? So, a review at the cell membrane. So this is sort of a phospholipid bilayer, and it's going to look like this. So the neuron phospholipid bilayer looks like every other phospholipid bilayer. It has a whole bunch of phospholipids. It's got cholesterol in there. It's got proteins hanging off of it. It's got channels. Some of the membranes, right, they have the neurolemocytes around, but the membrane itself is made up of like the same stuff that every other cell membrane is made of. Which means that things that can go through are the same. So hydrophobic molecules like fats can still move across. Small, uncharged molecules like oxygen can just go zipping right through. Carbon dioxide can just go zipping right out, right? Water can go in through those aquaporins, but really big molecules, they're gonna need some channels. And the ones that we're most interested in are these, the ions, because that's what we need to move to create that electricity, right? Anytime we move a charged particle, we create a little voltage. Now, this is where we're going to be working. So this is a multipolar neuron, a diagram of one that we would find in your brain. So every one of those dendrites can come in contact with a different neuron. Every one of them. So what that means is your cells are talking to each other all the time, okay? And they are making sure that everything in your body is running in homeostasis. Yeah, we haven't used that word in a long time. Thought I'd throw it in there, right? They are making sure that all of our negative feedbacks are working properly, okay? So what we are going to do is we are going to look at the electrical signals that are going to be moving along the cell membrane. So we're going to start by looking at the electrical signals. We're just going to do a little bit of this, then we're going to go in and slice a brain. Okay? So we already know, you already know that where the action is for an action potential is right here at the phospholipid bilayer. We have our outside, we have our inside, or ECF and ICF. Okay? Now, the membrane potential, or the possibility that something can happen, is dependent upon the ionic differences across the membrane. In other words, we need gradients. We need gradients. If you remember, on the outside, we have sodium, and on the inside, we have potassium. Now, it's okay that they are both positively charged. That's not the issue. The issue is, where are all the sodium and where are all the potassium? Because we have, if this is our setup, we have just created a sodium gradient from high on the outside to low on the inside. And we have created a gradient for potassium that is high on the inside and low on the outside. So this gives us the potential to do something, to send sodium down its gradient, to send potassium down its gradient. And when we do that, we change the membrane potential, right? From potential to actually doing something. And when that happens, we call that the action potential. The action potential. So, when we move sodium down and potassium down their gradients, that is what we call the action potential. And we are going to learn, not right now, next week, the sequence of events. What channels open, who goes down their gradient first, what channels close, what channels open, and all of that. But then the question is, why? Why would we want to change from just having the ability to do something to actually doing it? What are we trying to accomplish in the nervous system? Anything?
Homeostasis?
Yeah, we are trying to do homeostasis, but even more specific than that. That's kind of big picture. What about small picture? How are we going to do homeostasis? What do you want to use your nerves for? Or your brain?
To signal?
Yeah, to signal, to send signals. We want to be able to transmit or transfer information from one part of the body to the other part of the body. Because if we are not able to do that, we are not going to be able to maintain homeostasis. If you cannot tell your brain that you are cold and you are out on an iceberg, right? Your brain is not going to know to try and warm you up, and you will die, right? So if we can't get information from point A to point B in our body, all bets are off, right? Things are not going to work well. Does that make sense? Right? Okay, so that's where we're going to end for today. Let's get ready to go into lab by about 25 after. And then you need to make sure you have your goggles so that you can protect your eyeballs from brain parts. So, there you go.